other phylogenetic trees, right? And the idea is if we could find a way, either genetically or through protein similarities, amino acid sequence similarities, you should end up with one of these trees. Either humans are more closely related to gorillas than chimps and bonobos, or they're more closely related to chimps and bonobos than gorillas, right? Or they're an outgroup and the chimps and gorillas are more closely related to one another. So, based on these possibilities, people have been looking at all kinds of other genes to see if they can gain any insights into things. This particular one is based on the gene cytochrome oxidase 2. And again, you look at the sequences of A, C's, T's, and G's and see which sequences are more similar. And if you do that, looks like they built this tree using 6 human sequences, 5 common chimps, that's the pygmy chimp, and then 4 bonobos, and 6 gorillas, 3 orangutans, and 2 gibbons, that the human sequences are more similar to each other of these mitochondrial DNA sequences, and the bonobo and the chimp are more closely related because that's their common ancestor. Then there's the common ancestor of the human and the chimps, and then here's the common ancestor for the gorillas and the humans. So the African great apes cluster together based on cytochrome oxidase, humans being more closely related to chimps than they are to gorillas. Mitochondrial DNA is often used to do this kind of DNA work because unlike the DNA that's found in the nucleus, that during prophase 1 does all this crossing over and recombination, you don't get that with mitochondrial DNA, right? It's a haploid genome, so you don't get this recombination. There's no prophase 1. There's just a DNA replication step before binary fission. Okay? All right. All right, so if we take the cytochrome oxidase data and you take some protein data and you kind of combine all of this data together that's been used to give you a molecular clock, you get this tree, which is what's called a consensus tree. Right, a consensus phylogenetic tree. Consensus means that that's the one that, if you put all the trees together, this is the kind of common or consensus look to it. Currently, we accept that humans are more closely related to chimps based on DNA and protein sequences than we are to the gorillas or orangutans or gibbons or the old world monkeys. The red in here is kind of the plus or minus the standard error. So based on this, the human gorilla split, human chimp gorilla split, which would be right there. Here, the number for that is 6.4 million years ago, plus or minus 1.5 million. Right? Which lines up pretty well with our chart. And then the human chimp split, which would be right here, that split 5.4 million years ago, plus or minus 1.1 million years. Okay? And then some additional evidence has to do with looking at chromosomes. So humans have 46 chromosomes. And if you look at the chimps and gorillas, they have 48 chromosomes. So the other African great apes have 48 chromosomes. So then you ask yourself, well, how do you get from here to here? Well, during meiosis, you have one of those goof-ups. It's just instead of a goof-up with a non-disjunction, you have this weird, what's called a translocation. Translocations occur where tetrads get too close together. So I have a tetrad here, and normally there would be another tetrad over here during prophase 1, but one of the other tetrads gets too close, and now instead of crossing over and recombination occurring between homologous chromosomes, right, sister, non-sister chromatids, here I'm getting a crossover event, or here, between different chromosomes. And with this crossover, I could take a piece of this one and end up sticking it on the end of that one. If you take in the chimp and you put these two chromosomes together through this kind of weird translocation, you get our chromosome 2, which sequence-wise, this chromosome 2 is composed of a sequence that's similar to the African great apes chromosome here, plus this part of the chromosome is similar to their other chromosome. So we think you got from here to here through a weird fusion translocation, so maybe we're talking about one of those sympatric speciation events where changes in chromosome numbers made it so that the offspring, right, that lineage that comes to become humans, that original ancestor perhaps arose through a sympatric speciation event. Incidentally, changes in chromosome numbers is not all that uncommon in the mammals. The horse group that includes the zebras, horses, donkeys, and then there's all these weird kind of zebra-like things in Africa. They all differ from each other in like 1 or 2 chromosomes. So we think that meiosis, again, changes in chromosome numbers led to the diversification of the horse lineage. Okay? All right. All right. Now, we can also, in addition to looking at specific genes like cytochrome oxidase 2 or the protein serum albumin, we could look across large stretches of DNA. So instead of looking at a gene, let's look at a section of chromosomes. Since we share a lot in common with the other African great apes in terms of chromosomes, let's take a chromosome we have in common, maybe chromosome 5 in the human, and look at chromosome 5 in the chimp, and let's compare all of these sequences to see how much of a difference we've got between these two chromosomes. Is that okay? All right, so do you remember what we called differences in sequences? So if this is one of the sequences and this is the other sequence, and we had a 1 out of 5 difference, remember the term that we used to describe this? Right? We called this genetic distance. Genetic distance is, in this case, 20% or 0.2, because 1 out of 5 nucleotides is different between these two sequences. Okay? All right, so I'm doing okay on time. Rather than cut off really early for the semester, I thought I'd take off about 10 minutes for a lecture. So that's what I'm going to do right now, is go ahead and cut us at 45 instead of 55. And I'll see you next time. All right, take care.